In clinical trial management, there is a concept called a protocol deviation. It is defined as any departure from the approved study protocol that has not been reviewed and authorized in advance. Protocol deviations are not always catastrophic. Some are minor administrative errors that have no impact on the scientific validity of the data. You document them, report them, and move on. But some protocol deviations are what we call critical. A critical deviation is one that directly and materially affects the integrity of the trial data, the results on which a drug's safety and efficacy will be evaluated. A critical deviation that goes uncorrected and unreported to the FDA is not just a scientific failure. It is a federal crime. I had known about the critical deviation in the Veridex 7 trial data for 6 weeks before my boss stole my research. My name is Nora. For the previous 3 years, I had served as the senior clinical trial manager overseeing the phase 2 trial of Veridex 7, a promising new targeted therapy for a particularly aggressive form of non-small cell lung cancer. It was the most complex and consequential project of my career. I had designed the patient stratification model, managed the data collection across 6 participating hospital sites, and supervised the statistical analysis that had produced, after 3 years of painstaking work, a set of results that genuinely excited the oncology research community. The Veridex 7 trial data suggested a 31% improvement in progression-free survival compared to the current standard of care. In cancer medicine, a 31% improvement is not incremental. It is significant. It was the kind of number that attracts major pharmaceutical interest, generates massive grant funding, and propels careers into the stratosphere. My boss was Dr. Conrad Hale. He held the title of Deputy Director of Clinical Research at our institution. He was tall, effortlessly polished, and possessed the particular kind of confidence that comes not from deep competence, but from the successful performance of it, for long enough that nobody checked. He had a talent for entering rooms already in progress and summarizing the work of others as though it were his own contribution. He gave excellent presentations. He was extremely well connected within the pharmaceutical industry. He was not, however, a scientist in any meaningful operational sense. He hadn't personally managed a trial in over 7 years. He understood the broad outlines of clinical research, enough to speak intelligently at conferences, but he did not understand the granular, technical machinery of statistical methodology, patient stratification logic, or FDA regulatory compliance at the level required to actually build and run a phase 2 trial. This is important context for everything that followed. 6 weeks before my manuscript draft was finalized, I ran a routine audit of the patient data across all 6 trial sites. It was a standard procedure I performed quarterly. I cross-referenced the enrollment records, the treatment assignment logs, and the outcome data against the original stratification criteria defined in our FDA-approved protocol. The audit flagged an anomaly in Site 4, a hospital in Phoenix. The anomaly was subtle, the kind of thing that only reveals itself if you are performing exactly this kind of systematic, granular cross-validation. A software update to Site 4's electronic data capture system had been applied 7 months into the trial. The update had changed the way the system categorized a specific patient subgroup, patients with a prior history of immunotherapy treatment. Before the update, these patients were correctly being flagged in the system as prior immunotherapy and routed to the appropriate stratification arm of the trial. After the update, a coding error caused the system to recategorize them as no prior immunotherapy, silently routing them into the wrong treatment arm. 19 patients had been incorrectly stratified as a result of this error. In a phase 2 trial with our enrollment numbers, 19 miscategorized patients represented approximately 11% of our total sample. 11% of a sample being assigned to the wrong treatment arm does not just introduce noise into the data. It fundamentally alters the statistical validity of the entire dataset. The 31% improvement we had calculated in progression-free survival was not real. It was an artifact of the stratification error. The actual improvement, properly recalculated with the corrected patient assignments, was closer to 17%. Still promising, still publishable, but not 31%. Not the headline number that would launch careers and attract hundreds of millions of dollars in pharmaceutical investment. I documented the discovery meticulously. I wrote a detailed internal deviation report, timestamped in our secure electronic trial master file, describing the software error, the affected patients, the scope of the data corruption, and the corrected statistical outcome. I filed the report under my login credentials, with the date and timestamp embedded in the system metadata. Then I sat at my desk for a long time and thought about what to do next. The correct, unambiguous answer was straightforward. Report the deviation immediately to the Institutional Review Board, notify the FDA, correct the statistical analysis, and resubmit the revised results. This would delay publication by months, possibly a year. The corrected results were still scientifically meaningful. 17% was still a real finding worth publishing. But 17% does not generate the same excitement as 31%. I had been working on this trial for 3 years. I had been surviving on a mid-level researcher's salary, managing a team of 8 people, running a logistically complex, multi-site trial on a drug that I genuinely believed could help patients. I deserved the publication credit. I deserved the career advancement. I sent an email to Dr. Hale's assistant requesting a meeting to discuss an important data integrity issue I've identified that needs to be addressed before we finalize the manuscript. The meeting was scheduled for the following Thursday at 2 p.m. On Wednesday afternoon, Hale's assistant called to tell me the meeting had been rescheduled to the following week due to a conflict. On Thursday at 9:00 a.m., the morning of the meeting that had been rescheduled, I received an email from the administrative coordinator of our department's research publication office. "Dear Dr. Nora," it read, "wanted to let you know that the manuscript for the Veridex 7 Phase 2 trial results has been submitted to the New England Journal of Medicine this morning by Dr. Conrad Hale. Your name has been included in the author list. Please review the attached acknowledgement form." I opened the attached PDF. The author order was listed as follows: Conrad Hale, PhD, first author, followed by 5 additional researchers from our institution. My name was listed 4th. I was listed 4th on my own 3 years of work. But that was not the part that made my blood run cold. The part that made my blood run cold was the statistical results section, which I could see in the submitted abstract that was attached to the email. The progression-free survival improvement cited in the submission was 31%. The uncorrected, invalid 31%. The number that was generated by 19 miscategorized patients who had been silently routed into the wrong treatment arm by a software bug 7 months into the trial. Hale had taken my manuscript, changed the author order, and submitted it to the most prestigious medical journal in the world, with fraudulent clinical trial data inside it. He had no idea the data was fraudulent. He hadn't done the audit. He didn't know about the deviation report I had filed 6 days ago. He was not the kind of scientist who checked his own data at the granular level. He had read the summary, seen 31%, understood that 31% was career-defining, and submitted it without ever looking under the hood. I sat at my desk with the email open on my screen. I felt a specific kind of cold clarity that I associate with moments of very high professional stakes. I needed to make a decision. Option 1. I could send Hale an urgent email right now, describe the deviation, explain that the data was fatally flawed, and demand that he immediately withdraw the submission from NEJM. This was the ethical, correct, procedurally appropriate action. It would also immediately hand Hale the information he needed to save himself. He would withdraw the paper. The delay was due to rigorous data verification, correct the numbers, and resubmit under his own name, still first author. He would suffer no consequences. I would receive no credit. He would have successfully stolen 3 years of my work and suffered no consequences whatsoever. Option 2. I could say nothing. I pulled up my internal trial master file. I verified one final time that my deviation report was correctly timestamped, correctly filed, and correctly attributed to my login credentials. The metadata was clear and undeniable. I had documented the flaw 6 days ago. Any subsequent FDA investigation would be able to see with absolute certainty that I had known about the critical deviation before the manuscript was submitted, and that the corresponding author, Dr. Conrad Hale, had not. I closed the trial master file. I opened a blank email to Hale's assistant and typed a response to the publication acknowledgement form. Confirmed, I wrote. Thank you for including me. I sent the email. I closed my laptop. I went to get a cup of coffee from the break room. I had made my choice. The bomb was already ticking. I just needed to wait quietly, do my job, and let Conrad Hale walk into the blast radius of his own ambition. The New England Journal of Medicine does not publish quickly. The peer review process for a Phase 2 oncology trial result takes a minimum of 8 weeks, often longer. The journal sends the manuscript to 2 or 3 anonymous external reviewers, leading oncologists from other institutions, who scrutinize the methodology, the statistical analysis, and the clinical claims with a level of rigor that is supposed to catch exactly the kind of error buried in the Veridex 7 data. Supposed to. The problem with peer review is that reviewers evaluate what they are given. They read the manuscript. They examine the statistical tables. They assess the methodology as described. They do not have access to the raw underlying trial data. They cannot audit the patient-level records. They cannot check whether the numbers in the paper match the actual numbers in the original trial master file. A critical stratification error embedded in a complex, multi-site phase 2 dataset is invisible to a peer reviewer working only from the submitted manuscript. It requires a data auditor with direct access to the source files to find it. Exactly the kind of granular investigation that I had conducted, and that the external peer reviewers at NEJM were not equipped to perform. The manuscript passed peer review. 8 weeks after submission, the NEJM sent Hale an acceptance letter with minor revisions. The revisions were cosmetic, clearer labeling on 2 statistical tables, a minor adjustment to the limitations section. Nothing that touched the underlying data. Nothing that came close to the stratification error sitting in the patient records from Site 4 in Phoenix. Hale came into our department's weekly research meeting that Wednesday morning looking like a man who had just won a personal lottery. He announced the NEJM acceptance to the full team with the practiced modesty of someone who had been rehearsing the announcement for weeks. This is the result of tremendous collaborative effort from the entire team, he said, gesturing broadly to the room. His eyes swept past me without pausing. I'm incredibly proud of what we've accomplished together. The room applauded. People congratulated him. Someone produced a bottle of sparkling wine from somewhere, which struck me as deeply inappropriate for a Wednesday morning in a research department. But nobody else seemed bothered. I applauded too. I made sure to. The paper was published 6 weeks later. The NEJM publication of a 31% progression-free survival improvement for Veridex 7 in advanced non-small cell lung cancer was immediately picked up by oncology news networks, science journalists, and pharmaceutical industry analysts. Within 48 hours of publication, the story had appeared in the Wall Street Journal's health section under the headline, "Promising New Data Suggests Breakthrough in Aggressive Lung Cancer Treatment." 3 major pharmaceutical companies issued statements expressing interest in licensing conversations. The institution's communications department sent out a press release featuring a photograph of Hale, professionally lit, standing in the research wing, crediting him as the lead investigator. 2 weeks after publication, the institution's board of directors voted to create a new position, Director of Oncology Research Innovation. They named Conrad Hale as the inaugural director. The position came with a $40,000 salary increase, a dedicated research budget, a larger laboratory space, and a staff of 12. At the all-staff announcement meeting, Hale gave a speech about vision, about the future of precision oncology, about the responsibility of scientific leadership. He used the word integrity twice. I counted. He reassigned me the following Monday. He did not fire me. That would have been legally messy, and Hale was not a man who tolerated legal messiness. Instead, he simply restructured the research team. He promoted 2 junior researchers who were personally loyal to him into leadership roles on the institution's next clinical project. He moved me to a cross-departmental administrative support role, coordinating regulatory submissions, and managing protocol documentation for 3 separate, smaller trials that each had their own dedicated managers. It was not a demotion in title. It was a demotion in everything else. I went from leading the most significant oncology trial in the institution's recent history to processing paperwork for projects where my specific expertise was largely irrelevant. Hale accomplished this quietly, without confrontation, using the language of organizational restructuring and optimizing team strengths. It was textbook institutional bullying, the slow, invisible suffocation of a person's professional relevance. I moved into my new office, which was smaller and windowless, and I kept working. I also kept waiting. 3 months after the NEJM publication, I began doing something I had never done before in my professional life. I started reading the Veridex 7 publication citations obsessively. Every week, I searched the major oncology journal databases for any paper that cited the Hale et al. Veridex 7 study. I was looking for one specific thing: an independent replication attempt. In biomedical research, replication is everything. A single study, no matter how prestigious the journal, is considered preliminary evidence until it is independently confirmed by a separate research group working with their own patient population. Major pharmaceutical investment decisions, FDA approval applications, and changes to clinical treatment guidelines all require multiple independent confirmations of a finding. Hale's 31% was a single study. It would be replicated. The question was when and by whom. The answer came 7 months after publication, in the form of a brief correspondence published in the Journal of Clinical Oncology. The authors were a group of oncology researchers from the University Hospital in Freiburg, Germany. The title of the correspondence was Measured and Clinical, the way scientists write when they are being politely devastating. Failure to replicate. Reported. Progression-free survival benefit in Veridex 7 Phase 2 trial. Preliminary observations from an independent cohort. I read the full text in my small, windowless office with the door closed. The German group had conducted their own prospective evaluation of Veridex 7, using a nearly identical patient population and a rigorous stratification protocol. Their results showed a progression-free survival improvement of approximately 16% compared to standard care. 16%. My corrected number had been 17. The German group had landed within 1 percentage point of the truth. They were careful in their language. As scientists always are when challenging a peer-reviewed publication, they did not accuse Hale of fraud. They noted that the discrepancy between their 16% and the published 31% was statistically significant and warrants further investigation, and they called for an independent data audit of the original trial records. I saved the paper to my personal drive. I made a printed copy, date-stamped, and placed it in a physical file in my locked desk drawer, alongside the printed copy of my internal deviation report with its timestamp from 11 months ago. I then did the specific thing that every clinical trial regulation specialist knows to do when independent replication fails to confirm a published result. Nothing. I did not contact the German researchers. I did not call the FDA. I did not send Hale a copy of the German paper with a taunting note attached. I simply waited for the machinery to turn. Because I knew, with absolute regulatory certainty, what happens next when an NDJM published, FDA scrutinized, high-profile phase 2 oncology trial result fails to replicate at a reputable independent institution. The FDA does not wait for an invitation. They open an investigation on their own authority. Within 6 weeks of the German publication, I expected a call, and I knew exactly what I was going to say when it came. What I hadn't fully anticipated was what Hale would do when he saw the German paper. He summoned me to his new, larger office, the one with the window overlooking the research courtyard. On a Tuesday afternoon, his assistant told me it was a quick check-in. Hale was standing at his window when I walked in, which I had noticed was a power positioning choice he had recently adopted. He gestured for me to sit. He remained standing. "I've just been made aware of a correspondence in JCO from a German group," he said, his tone carefully casual. "A replication attempt that shows a lower efficacy number than our published result." He turned from the window. "Were you aware of this?" "I read it when it was published," I said. "And you didn't bring it to my attention immediately?" His voice had an edge of reproach. "It was published correspondence in an open-access journal," I replied evenly. "I assumed you had journal alerts set up for citations of your own published work." A very slight tightening around his jaw. He hadn't. He had been too busy giving conference presentations and meeting with pharmaceutical licensing teams to monitor citations. "The German result could be explained by population differences," he said, redirecting quickly. "Different genetic backgrounds, different standard of care benchmarks. I'm going to ask our statistician to draft a response letter to JCO explaining the methodological context." I said nothing. I waited. "Nora," Hale said, sitting down now, leaning forward slightly, "is there anything in the original Veridex 7 dataset that you think could be problematic? Anything you noticed during the trial that might explain a discrepancy of this magnitude?" He was fishing. He could feel the ground moving beneath him, but he didn't know why, and he was hoping I would tell him. I looked at him across his expensive new desk. I thought about my 3 years. I thought about my windowless office. I thought about the timestamped deviation report sitting in my desk drawer. "The data was collected and managed according to the approved protocol," I said, which was precisely true and precisely uninformative. "If there are questions about the statistical methodology, those would be best addressed by a full data audit." I watched him absorb this. I watched him decide whether full data audit was a threat or a reassurance. He couldn't tell. His face was very still. "Right," he said. "I'll have our statistician look into it." He dismissed me. I walked back to my small office. I sat down. I opened my email and checked my institutional inbox, my personal inbox, and my registered FDA contact email address. 3 days later, the FDA contact email had a message in it. Subject line: "Notice of data audit, Veridex 7 Phase 2 clinical trial, ORI case number 2024-0847. The machinery had turned. The bomb had gone off. And I had not touched a single wire. The FDA's Office of Research Integrity does not send a polite letter and wait for a convenient time to talk. When they open a formal data audit on a published clinical trial, they move with the methodical, invasive thoroughness of a federal agency that understands exactly what is at stake when fraudulent medical data enters the scientific literature. The audit notice informed our institution's Research Compliance Office that a team of FDA data auditors would be arriving on site in 14 days to conduct a full review of all source documents associated with the Veridex 7 Phase 2 trial. They requested that all trial master files, patient-level data exports, site monitoring logs, electronic data capture audit trails, and protocol deviation records be made available in their original, unaltered digital format. The words unaltered digital format and audit trails are the two phrases that every person involved in fraudulent research data dreads most. Electronic trial management systems automatically record every action taken within them. Every data entry, every modification, every file access, every deviation report filed. The audit trail is a comprehensive, tamper-proof digital ledger that shows exactly who did what and when. You cannot retroactively alter a time-stamped audit trail in a validated clinical data system without leaving evidence of the alteration itself. The system records the alteration. It is one of the foundational principles of FDA-compliant data management. Hale did not know this with sufficient granularity to understand the specific, terminal nature of his situation, but he understood that an FDA audit was catastrophic. And when cornered, Hale's default operating mode, the one I had watched him deploy in every difficult institutional situation over the 4 years I had worked under him, was to find someone else to stand in front of the firing squad. He called me into his office the morning after the audit notice arrived. This time, he was not standing at the window with practiced authority. He was sitting behind his desk, and his posture was slightly wrong. Too upright, too controlled, the specific physical stilts of a man managing panic. "I've spoken with our legal team this morning," he said. "As you know, the FDA has initiated a data audit. Given that you were the operational lead on the Veridex 7 trial, they are going to want to speak with you directly." "Of course," I said. "I'll cooperate fully." "Yes," Hale said. He paused. He was constructing a sentence very carefully. "The legal team wants to make sure everyone is aligned on the narrative around the trial conduct, specifically regarding the data management at Site 4. There were some complexities there." This was the moment I had been waiting for and had thoroughly prepared for. "There were complexities at Site 4, yes," I said calmly. "I documented them in an internal deviation report filed 11 months ago. It's in the trial master file." Hale went completely still. "A deviation report," he repeated. "A critical protocol deviation," I said, keeping my voice entirely even. "A software update at Site 4 created a patient stratification error affecting 19 subjects. I identified it during a routine quarterly data audit. I scheduled a meeting with you to discuss it. But the meeting was rescheduled, and before I could reschedule again, the manuscript had already been submitted to NEJM. The silence in the room was the loudest I had ever heard in a professional setting. I watched the specific sequence of realizations move across Hale's face. First, the understanding that the German replication failure was not a random statistical anomaly, but a direct consequence of a documented error he had never been told about. Second, the understanding that this error had been documented. Tepstürzt, formally filed, attributed to a named investigator, before he had submitted the flawed manuscript. Third, the understanding of what this meant for him, personally, when the FDA auditors pulled the trial master file and found that document. He had submitted a manuscript containing a known critical protocol deviation, known to the person who conducted the trial, who had filed a formal internal report, who had scheduled a meeting to inform him, and who had been stood up. The paper had been submitted without the deviation being corrected or reported to the FDA, as required by federal regulation. He was the corresponding author. The corresponding author is legally and professionally responsible for the integrity of the submitted data. I could see him searching for the exit. I could see the massive intelligence of his self-preservation instinct engaging, looking for a way to construct a narrative in which this was my failure rather than his. "Why didn't you escalate when the meeting was rescheduled?" he said, his voice finding a harder edge. "If you identified a critical deviation, you had a regulatory obligation to escalate immediately. If you sat on this information..." "I have the email I sent to your assistant requesting the meeting," I said. "I also have the deviation report in the master file with my login timestamp. And I have the acknowledgement email I received from the publication office, sent the morning after our meeting was rescheduled, confirming the manuscript had already been submitted. The timeline is documented." Hale stared at me. He was not a stupid man. He understood that I had just described a paper trail that ran directly between his decision to steal my research and the fraudulent publication that now had his name on it. "You knew the data was wrong," he said, very quietly. "When you got that acknowledgement email and saw the submission had gone through, you knew." "I knew there was a critical deviation in the dataset that had not been corrected," I said. "I have documented proof of when I knew it and how I attempted to report it." "And you said nothing." His voice was almost a whisper. "You let it go to publication knowing the data was compromised." "I sent back the author acknowledgement form," I said simply. "Fourth author, as listed." He stared at me for a very long time. I held his gaze without difficulty. I had nothing to be afraid of. My hands were clean, my timestamps were clean, my deviation report was clean. The only thing I had done was fail to bang loud enough on a closed door. And the reason that door was closed was because the man sitting across from me had canceled our meeting, so he could submit my research under his own name. "Get out," he said. I stood up. I smoothed my jacket. I walked to the door. "Nora," Hale said. I stopped. I turned. "If you think this is going to go the way you're imagining," he said, his voice low and controlled, "you should know that I have the resources and the institutional relationships to make your professional life extremely difficult. I have friends on the review board. I have relationships with every major pharmaceutical sponsor in this field. You will never lead another trial if I decide you shouldn't. I considered this for exactly as long as it deserved. Then I suppose it's fortunate," I said, "that I have a signed, timestamped deviation report and you have a fraudulent NEJM publication." I left. He was not bluffing about his institutional relationships. Within 3 days, I received an internal memo informing me that my access to the primary research data systems had been placed under temporary administrative review pending institutional compliance assessment. It was a lockout. He was trying to prevent me from accessing any additional data before the FDA arrived. But the specific data I needed, my deviation report, my original audit logs, my email to his assistant requesting the meeting, was already secured. I had printed hard copies. I had saved encrypted backups to a personal drive. And I had saved an encrypted copy to a personal cloud account maintained under my individual professional credentials rather than the institutional system. 3 days before the FDA auditors arrived, I received a call from the institution's legal counsel. They wanted to meet with me to discuss my role in the Veridex 7 audit process. This is standard procedure. Institutions always want to debrief their staff before a federal audit. I arrived at the legal team's conference room with my own personal attorney. She had spent 12 years specializing in research whistleblower law and FDA regulatory compliance before going into private practice. I had retained her 2 months before the German paper was published because I had known this day was coming and I had known I would need someone in my corner when it did. Hale's people had been counting on me walking into that meeting alone. The look on the institution's general counsel's face when my attorney sat down beside me told me everything I needed to know about how they had expected the conversation to go. My attorney laid a copy of the time-stamped deviation report on the conference table. She laid a copy of the email to Hale's assistant. She laid a copy of the publication acknowledgement email with its timestamp showing it was received after the meeting was canceled. and she laid a single, very clean cover letter citing the relevant FDA regulatory statutes and my full documented cooperation with the incoming audit. My client documented and attempted to report a critical protocol deviation before the manuscript was submitted, my attorney said. She will cooperate completely with the FDA data audit. She will provide all documentation on request. She has no exposure in this matter. She paused. The same cannot be said for the corresponding author. The institution's legal team looked at the documents. They looked at each other. They asked for a recess. When they came back 20 minutes later, the general counsel's tone had shifted entirely. They were no longer treating me as a target. They were treating me as a witness. The FDA auditors arrived on Monday morning. I spent 4 hours in a conference room with 2 of them, walking through the trial master file, the deviation report, the site monitoring logs from Phoenix, and the electronic audit trail showing every data entry and modification across the entire lifespan of the trial. I answered every question completely, accurately, and without editorializing. I pointed to the deviation report when they asked about the stratification error. I showed them the timestamp. I showed them the meeting request. I showed them the acknowledgement email. I did not need to accuse Hale of anything. The documents accused him entirely on their own. At 4:30 p.m. on the second day of the audit, one of the FDA investigators closed her laptop and looked at me with an expression that was professional and carefully neutral, but behind which I could read the specific exhaustion of someone who has spent 2 days confirming something they already suspected. "Dr. Nora," she said, "is there anything else you'd like to add to the record?" "No," I said. "I think the record is complete." She nodded. She thanked me for my cooperation. She picked up her documents and left. I sat alone in the conference room for a moment. Through the window, I could see the research courtyard Hale's new office overlooked. It was a gray afternoon. The courtyard was empty. I thought about 3 years of trial management. I thought about 19 patients in Phoenix being silently routed to the wrong treatment arm by a software update that nobody caught because everyone was exhausted and underfunded and trying to do the work of 12 people. I thought about a 31% number that was never real. Then I picked up my bag and walked out. I had told the truth. The FDA would do the rest. The FDA's formal investigation of a published clinical trial operates on 2 parallel tracks simultaneously. The first track is the scientific track. A complete granular audit of all source data, audit trails, and protocol documents to determine precisely what went wrong and how severely it affected the validity of the published results. The second track is the regulatory track, a determination of who is legally and professionally responsible for the integrity of the data under the federal regulations governing clinical trial conduct and what the appropriate enforcement action is. These 2 tracks converged on Conrad Hale with the slow, grinding inevitability of 2 tectonic plates. The scientific audit took 4 weeks. The FDA auditors had full access to the electronic trial master file, including every audit trail entry across all 6 trial sites. They pulled the Site 4 electronic data capture logs and confirmed exactly what my internal deviation report had described: the software update, the miscategorization of 19 patients, and the downstream effect on the stratification analysis. They then reran the core statistical analysis using the corrected patient assignments. Their independent calculation matched mine within half a percentage point, a 17.3% improvement in progression-free survival, compared to the 31% published in the NEJM. The FDA issued a formal finding of significant departure from protocol, the agency's designation for a critical deviation that materially compromised the scientific validity of the trial results. They also found my deviation report, the timestamp, the meeting request email, the chain of documentation. showing that the deviation had been identified and an attempt had been made to report it before the manuscript was submitted. And they found something else I had not anticipated, which refined the severity of their finding considerably. The Site 4 software update, the one that had caused the stratification error, had generated an automatic alert in the electronic data capture vendor's system at the time it was applied. The alert had been routed to the trial's principal investigator of record. The principal investigator of record on the FDA's approved protocol was not me. Operationally, I ran the trial, but the formal regulatory submission to the FDA that had approved the trial listed Hale as the principal investigator because Hale's institutional title and his relationships with the pharmaceutical sponsor had been used to secure the trial approval. He had attached his name and credentials to the trial at the beginning to access the funding and the prestige. And in doing so, he had assumed, under federal regulation, the legal responsibility of the PI for all regulatory compliance matters. The automatic vendor alert about the software update had been sent to his institutional email address 14 months ago. His email system had auto-sorted it into a subfolder for trial management administrative correspondence. He had never read it. This meant that the FDA could establish, with documented evidence, that the principal investigator had received notification of a potential data system issue at Site 4 and had taken no action. This elevated the finding from critical deviation with inadequate reporting to a much more serious designation: failure of principal investigator oversight, resulting in submission of materially compromised clinical data to a peer-reviewed journal. In FDA regulatory language, submitting clinical trial data that you knew or should have known to be compromised, especially when you are the PI of record, is the most serious category of research misconduct the agency can act upon. The second finding compounded the first. When the auditors reviewed the manuscript submission correspondence, they found the email chain showing that Hale had requested and received my completed manuscript draft, then submitted it to NEJM without conducting any independent verification of the underlying data integrity. As the corresponding author and PI of record, he was required under FDA regulations to certify the accuracy and completeness of the data before submission. His submission email to NEJM had included this certification. He had certified the accuracy of data he had never audited for a trial he had not operationally managed after receiving a software alert he had never read. The FDA issued its preliminary findings letter 6 weeks after the audit concluded. The letter was addressed to Hale personally, with copies to the institution's Research Compliance Office, the NEJM editor-in-chief, and the pharmaceutical company that had been negotiating licensing terms for Veridex 7. I received a copy 3 hours after Hale did, through my attorney. I read it at my kitchen table that evening. The findings letter was clinical and precise, as FDA documents always are. It described the deviation, the corrected analysis, the PI notification failure, and the submission certification issue. At the bottom of the findings section, it contained the phrase that constitutes the most severe consequence in academic research medicine. The Office of Research Integrity is initiating proceedings for permanent debarment of Conrad Hale, Ph.D., pursuant to 21 U.S.C. 335A, on the basis of findings of material falsification or gross negligence in the conduct and reporting of clinical investigation. Permanent debarment. In the FDA's enforcement framework, permanent debarment is reserved for the most serious category of research misconduct. A permanently debarred individual is prohibited, for the remainder of their career, from participating in any capacity in any clinical trial, drug development application, or regulatory submission that falls under FDA jurisdiction. They cannot serve as a PI. They cannot serve as a co-investigator. They cannot serve as a consultant. They cannot sign or submit any document that requires FDA-recognized credentials. In practical terms, for an academic research physician whose entire professional identity and income is built on FDA-regulated clinical trial work, permanent debarment is a complete and irreversible professional death sentence. The institution acted within 24 hours of receiving the FDA findings letter. They had no choice. Hale's employment was terminated. The Director of Oncology Research Innovation position, which had existed for 11 months, was eliminated. The institution issued a public statement acknowledging the FDA findings, expressing full cooperation with the regulatory process, and announcing the launch of an internal review of all clinical research oversight procedures. The NEJM issued a retraction notice for the Veradex 7 paper the following week. A retraction notice in the New England Journal of Medicine is one of the most public and professionally devastating events that can befall a researcher. The journal posts the notice permanently on the paper's page, visible to every person who ever searches for it. The notice specified critical protocol deviation resulting in material misrepresentation of efficacy data as the basis for retraction and named Hale as the corresponding author. The 3 pharmaceutical companies that had been in licensing discussions for Veradex 7 withdrew from negotiations immediately. The drug itself, which was a real compound with genuinely promising, if more modest, therapeutic potential, would have to begin the long process of rebuilding its evidence base under new investigators, starting from the corrected 17% finding. It would be years before it had a realistic path to FDA approval. Hale's pharmaceutical industry relationships, the ones he had threatened to weaponize against me, evaporated with his debarment. Nobody in the industry maintains professional relationships with a debarred investigator. The liability is too significant. I heard through institutional channels, several weeks later, that he had attempted to secure a position at a private research consulting firm, trading on his pre-debarment publication record. The firm had run a standard FDA debarment database check as part of their due diligence process, which every responsible research organization does as a matter of routine, and had immediately rescinded the offer. The FDA debarment database is publicly searchable. His name would be in it forever. 3 months after the debarment proceedings were finalized, my attorney received a letter from Hale's personal attorney. It was a settlement offer. Hale was proposing to pay me a confidential sum in exchange for a signed agreement that I would not pursue any additional legal action and would not make public statements attributing the Veradex 7 data errors to his conduct. My attorney and I discussed the offer for approximately 15 minutes. No, I told her. Are you sure? She asked. The number they're proposing is significant. He is offering me money to tell the scientific community that the fraud was ambiguous, I said. I am not interested in ambiguity. She sent back a declination. I did not pursue any additional legal action. I did not need to. The FDA finding, the retraction notice, the debarment, and the termination had already accomplished everything I needed them to accomplish. The record was clear. The record would remain clear permanently. Every researcher who ever searched Conrad Hale's name in the future would find the debarment, the retraction, and the FDA findings letter. His academic reputation was not damaged. It was erased. I did not feel the specific, sharp satisfaction I had expected when I first made the decision to stay silent 11 months earlier. What I felt was quieter than satisfaction. It was the specific, settled feeling of a person who had trusted a rigorous system to do what it was designed to do, and had been proven right. The system worked. Not because I had manipulated it, but because I had documented the truth carefully and let the machine find it on its own schedule. I went back to the full research database the following morning and started looking for a new trial to lead. The Veradex 7 paper was in the scientific literature for 14 months before the retraction notice appeared. 14 months during which oncologists in academic hospitals across 3 continents had read the 31% figure and discussed it in tumor boards and research conferences. 14 months during which 2 smaller institutions had begun preliminary planning for their own phase. 3 trials based on the phase 2 results. 14 months during which 1 pharmaceutical company had advanced its internal development program for Veridex 7, far enough to begin drafting a preliminary FDA approval application. None of these efforts resulted in a patient receiving Veridex 7, based on the fraudulent data. The drug had not yet reached a stage where it was being prescribed outside of controlled trial settings. The retraction came before any of the larger downstream decisions were finalized. I tell myself this. I tell myself it carefully and specifically, because it matters. No patient was harmed by the 14 months. The system caught the error before it propagated into clinical practice. This is the best possible outcome of a bad situation. But I also know, with the precision of someone who has spent a career thinking in data, that no patient was harmed is a statement about what happened, not about what could have happened. The gap between what happened and what could have happened is the space I live in now. My career after the Veridex 7 matter improved significantly, and in ways I had not entirely anticipated. The institution, in the aftermath of its legal exposure and its public compliance statement, commissioned a comprehensive internal review of its clinical research oversight procedures. They hired an outside firm to conduct the review. The outside firm's recommendation was that the institution needed a dedicated director of research data integrity, a new position specifically focused on building the kind of systematic, granular audit infrastructure that would have caught the Site4 software error before it metastasized into a published fraud. They offered me the position. The salary was a 30% increase over what I had been making in my windowless administrative role. The authority was substantial. I would have direct oversight of all data management protocols across every active clinical trial, with the explicit mandate to build an audit infrastructure robust enough to survive FDA scrutiny at any time. I accepted. In the 18 months since I took the new role, my team has identified 3 significant protocol deviations across the institution's trial portfolio. All 3 were caught early, corrected, and reported to the appropriate regulatory bodies in the timeline required by federal guidelines. None of them resulted in a published error. None of them required an FDA enforcement action. This is the work. This is what the job is supposed to look like when the infrastructure is functioning correctly. Hale's position, the Director of Oncology Research Innovation, the one he had occupied for 11 months before his termination, was quietly eliminated and never refilled. The institution absorbed his staff back into existing teams and redistributed the research budget to existing programs. The last information I have about Hale personally came from a former colleague who encountered him at a conference 8 months after the debarment. He was attending as a general registrant, not as a presenter or panelist. He was networking aggressively, trying to position himself for advisory or consulting work in adjacent industries where FDA debarment rules do not apply: medical device development, nutritional supplement research, healthcare technology. He still had his PhD. He still had his polish. He still knew how to walk into a room and perform authority. But in every room that mattered in his field, his name was now preceded by a permanent retraction notice and followed by a publicly searchable FDA debarment record. The performance had nowhere left to land. I have thought extensively about the ethical dimensions of what I did. I have thought about it the way I think about a dataset with a confirmed error. Systematically, without sentimentality, trying to identify exactly where the contamination entered and what it actually affected. The most honest accounting I can give you is this. I made a deliberate choice to allow a document containing a known critical error to be published in the most prestigious medical journal in the world. I made this choice because the document had been stolen from me by the person who would have needed to correct it. I made this choice knowing that the scientific and regulatory process would eventually identify the error, as it is designed to do, and that the consequences for the person responsible would be terminal. I do not believe I did a good thing. I believe I did a calculated thing. A good thing would have been to report the deviation through every available channel loudly and persistently, regardless of whether my career benefited or suffered as a result, regardless of whether Hale was held accountable or walked away free. A good thing prioritizes the integrity of the scientific record above everything else, including personal justice. I did not do that. I prioritized justice, personal, specific, pointed justice against a man who had stolen 3 years of my work, above the early correction of a scientific error. The distinction matters. I need to be honest about it. What I can also tell you honestly is this. Hale would have corrected nothing, even if I had sent him 100 emails and knocked on every door in the institution. He would have hired lawyers, delayed, minimized, and deflected until the paper was in the literature and his promotion was secured. He would have found a way to make the deviation correction look like a minor technical footnote, and he would have retained both the credit and the career. I cannot prove this. It is a judgment about his character based on 4 years of close observation. It may be the story I tell myself to make the calculation feel cleaner than it was. I hold both of these things simultaneously. The possibility that I was right about what he would have done, and the certainty that my silence was motivated, at least partly, by the desire to watch the machine destroy him, rather than the desire to protect the scientific record. I am a data integrity specialist. My entire professional life is built on the principle that the record must be accurate, complete, and honest. I allowed an inaccurate, incomplete record to exist in the scientific literature for 14 months because doing so served my interests. I live with this. I do not resolve it. I add it to the file and I go back to work. The Veridex 7 compound itself has a future, as it turns out. A new research team at a separate institution took on the project after the retraction. Starting from the corrected 17% finding, 17% is real and it is meaningful. Their preliminary Phase 3 design was approved by the FDA 6 months ago. They listed me as a scientific consultant on the approved protocol with full credit for the original trial design and the data methodology. My name is on that document correctly. It is a small thing. It is also the thing I look at when I need to remember what this was actually for. I know there are people who will say that what I did was unforgivable. They will say that a person of genuine integrity, a real scientist, someone who actually believes in the principles she claims to uphold, would never have allowed a flawed paper to enter the literature. They will say that the 14 months, the 2 institutions that began planning Phase 3 trials based on fraudulent data, the pharmaceutical development work that had to be discarded. All of that represents a cost to science that cannot be justified by one person's desire for professional revenge. They are not entirely wrong. There are also people who will say that I was entirely justified. They will say that Hale stole my research, removed me from my own work, placed me in a windowless office, threatened my career, and attempted to obstruct a federal audit. They will say that the system worked exactly as it was supposed to, and that I merely stepped aside and allowed it to function. They are also not entirely wrong. The truth, as it usually is, occupies the uncomfortable and specific territory between these two positions. I occupied that territory for 11 months, and I would be lying if I told you it was clean, so I will leave the verdict where I always leave it, with you. If your boss stole 3 years of your research, published it under his own name, and was weeks away from a career-defining promotion, Would you tell him about the fatal flaw in the data? Or would you file the deviation report, timestamp the evidence, and wait quietly in your windowless office while the FDA turned the key? Tell me honestly in the comments. I read every single one.